In promising new developments for treating ALS, the biotech company Neurizon Therapeutics has found a new use for an existing drug which was initially used as a deworming agent for treating livestock called Monepantel, now the active ingredient in NUZ-001. Monepantel is now being redeveloped for human use upon the discovery of its potential to slow the disease's progression and extend patients' lives with its unique properties related to cellular autophagy and protein clearance. Neurizon Therapeutics has obtained exclusive rights to adapt and develop Monepantel for human use, with formulation changes and safety optimization for human dosing. The primary challenge for ALS researchers has been finding how to stop or slow disease progression. In this search, researchers study autophagy, the body's natural process of cleaning out damaged or unwanted parts of cells to keep them healthy, including the clearing out of built-up proteins which are believed to form from cellular stress or nutrient deficiency. In ALS, this process of cellular recycling doesn't work properly, causing harmful proteins to build up inside nerve cells, which cause motor neurons, which control muscles, to die off. This results in a progressive deterioration of function, paralysis, and in the end stage, respiratory failure. NUZ-001 gives hope to slowing down this progression by blocking a cellular pathway called mTOR, a key role in regulating autophagy, which helps restore cellular recycling and reduces protein buildup, which could potentially slow down the progression of the disease. Significant clinical benefits across function, lung stability, biomarkers, and survival were found in a long-term study during which patients received a daily administration of NUZ-001, some for over 2.5 years. Patients who had been treated's motor abilities declined 31% slower than comparable historical patients on the ALS Functional Rating Scale revised, and their lung function, a key measure of the disease's progress, dropped 43% slower. The most striking finding was in survival rate. On average, patients lived about 16 months longer, and their risk of death fell by roughly 77 to 79%. Notably, the study showed the drug to be well tolerated, with no serious side effects reported throughout the treatment period. Researchers additionally looked at specific biomarkers in the patient's blood and urine to see what was happening on a molecular level. One of the biomarkers, called plasma neurofilament light chain, is a protein released when neurons are injured, remained steady over the course of therapy, while another biomarker, which signals ongoing nerve breakdown, actually declined. This reduction is interpreted as a sign that nerve damage is slowing or potentially reversing in ALS. Additionally, NUZ-001's therapeutic potential directly addresses the abnormal protein behavior of TDP43. This protein helps carry out genetic instructions inside the cell, which in ALS leaves the nucleus and enters the main body of the cell, forming harmful clusters that disrupt nerve cell function, and is found in approximately 97% of ALS cases. Preclinical studies indicate that NUZ-001 can clear these clusters, helping to restore the health and function of affected motor neurons. These findings strengthen the case for NUZ-001's further testing in the upcoming HEALEY ALS platform trial, a large multi-treatment study led by Massachusetts General Hospital, testing several promising therapies at once within one coordinated study, with the potential to dramatically accelerate the development of new ALS treatments and bringing us closer to a cure.